we will be starting the new chapter, that is sexual reproduction in flowering plants. Sexual reproduction in flowering plants. So we will start this chapter with a brief introduction. Flowers are object of aesthetic, ornamental, social, religious, and cultural value. Okay, they have always been used as a symbol for conveying important human feelings such as love, affection, happiness, etc. Okay, so now we will move on to the main topic. First, we will brush up the various parts of flower. Okay, here in this chapter, mainly we will deal with two parts of flower, that is pistil and stamen. This is here it is pistil and this is stamen. Okay, so pistil basically contain stigma. This is stigma, then style and ovary. Okay, I'll draw this for you. Okay, as this both part is important, so I want you to clear with this at least. So this is stigma, this is style, ovary, okay, and these three forms pistil. Okay, next is stamen. Stamen is actually more simple to understand. Okay, this is anther, this is filament, and whole together this is called as stamen. Okay, so now we will start with the longitudinal section of flower. This is actually longitudinal section of flower. You can refer the various part. I have already explained the two most important part we will be dealing in this chapter, that is pistil and stamen. Okay, so in male reproductive structure, basically androecium. Androecium is the male reproductive structure of a flower and gynoecium is a female reproductive structure of a flower. And androecium, it consists of stamen and it represent male reproductive organ. Okay, that's why I told you this is important, because here we are dealing with the reproduction part. Okay, so we will be mainly focusing on stamen and pistil. Okay, this is a stamen. Okay, this is, it represent male reproductive organ. Okay, stamen represents male reproductive organ. Okay, see, this is anther, this part is filament. Okay, and filaments, see, this is anther and this is attached to this part which is called as thalamus. Okay, I told you, just now I told you, stamen has two parts, anther and filament. Okay, anther has two part, sorry, stamen has two part. really sorry. Stamen has two parts. Stamen has two parts, anther and filament. So in this topic especially, which I'm going to cover now, in this topic we'll be dealing with the male reproductive part. Male reproductive part of a flower. Okay, so we need to focus on stamen, which has two parts, anther and filament. Okay, so stamen filament is long cylinder stalk like structure. Filament is, this is filament, long and cylinder stalk like structure, this is filament, and this is terminal bilobed structure. On top of it, it has terminal bilobed. Bi means two. So two lobe is present. Okay. This part is called as anther. This part is called as anther. Okay. Clear? And proximal end of filament. That this proximal end of filament, this is proximal end. This is attached to thalamus. Here you can see this is attached, this part is attached to thalamus. Okay, this part is attached to thalamus. Clear? Next we will move on to anther. Okay, so this is a diagram for anther. This is a diagram of anther. Okay, I'll explain this part now. So a typical angiosperm anther is bilobed. Okay, that is bilobed. I told you bilobed means two lobe, and each lobe is having two theca. Okay, each lobe is having two theca. Here you can see the theca. Here you can see theca. This is one theca. This is another. Okay, so this is anther. This is theca. Okay, and the longitudinal grooves runs lengthwise separating the theca. Okay, this is which is called as line of dehiscence. Here it is, line of dehiscence. Okay, a longitudinal groove runs lengthwise separating the theca. Okay, this is the basic structure of anther. Okay, this is a basic structure of anther. Now we will see the more enlarged structure of anther. So anther is four-sided. Okay, anther is basically anther is four-sided. Here you see anther is four-sided. Four-sided. Okay, anther is four-sided structure consists of four microsporangia. Okay, consists of four microsporangia, and all microsporangia is located at the corner. Corner. Two in each side. Okay, two in each lobe. So anther is four-sided. Anther is four-sided tetragonal structure. Tetragonal structure. It has four microsporangia. It has four microsporangia, and each microsporangia is located in each lobe. Basically, one in each lobe. Okay, and this microsporangia. Okay, this microsporangia develop further and become pollen sac. Okay, this microsporangia develop to form pollen sac. Okay, next we'll move on to microsporangia. Okay, assuming that you all have clear concept of male flower reproductive structure. Okay, so here we will be seeing a structure of microsporangium. This is a structure diagram for a structure of microsporangium. Okay, so this is a typical microsporangiums which appear circular in outline. Okay, I have drawn only 1/4 of it. Okay, you can complete it in a similar fashion. Okay, so typical microsporangium, if you will see, it appears circular in outline. Okay, it appears circular in outline. What I have drawn is only 1/4. You can finish this diagram in a similar fashion. You will get this circular shape. Okay, it is microsporangium is having four wall layer. It is having four wall layer: epidermis, endodermis, middle layer, and tapetum. Okay, it is having four layer: epidermis, endothecium, middle layer, and last one is tapetum. Okay, outer layer perform the function of protection and help in dehiscence of anther to release the pollen. Okay, so first one is epidermis, second is endothecium, then middle layer, and the last one is tapetum. This is the sequence, outer to inner. Okay, so outer layer, that is epidermis. Okay, it perform the function of protection. Okay, and help in dehiscence of anther to release pollen. Okay, innermost layer is tapetum. This is innermost layer. Innermost layer. Okay, I'll show you once. This is tapetum, innermost layer. Okay, this is tapetum. So cells of tapetum, if you will see, cells of tapetum has dense cytoplasm. Okay, it has dense cytoplasm. dense cytoplasm, and it is multinucleated. And it is multinucleated. It has more than one nucleus. Okay, more than one nucleus. And the function of tapetum is to nourish the developing pollen grain. Okay, it helps in nourishment of developing pollen grains. Okay, clear? So next we will be moving to microsporogenesis. Okay, next we will be moving to the process of microsporogenesis. Microsporogenesis. Process of formation of microspore. Process of formation of microspore from a pollen mother cell through meiosis. Through meiosis is called microsporogenesis. Okay. Process of formation of microspore from a pollen mother cell PMC through meiosis division is called as microsporogenesis. Okay, so I already explained you that anther, okay, and filament. This is anther, this is filament, both form stamen. It represents the male reproductive part, okay, part of flower. As you will go deeper, okay, so anther develops. When anther develops, okay, anther develops, cells of sporogenous tissue undergo meiotic division. Okay, sporogenous tissue, sporogenous tissue undergo division and form microspore tetrad. Form microspore tetrad. I showed you that, right? Microspore tetrad, which contain microsporangium. Okay. Then each cell of sporogenous tissue, each cell of sporogenous tissue is capable of giving rise to microspore tetrad. Okay. See. Anther develop. Okay. This is anther. Okay. Anther develop the cells of sporogenous tissue. Okay. Sporogenous tissue gives rise to microspore tetrad. Microspore tetrad. Okay, this is microspore tetrad, and each one is potential pollen or microspore mother cell. Okay, each cell of sporogenous tissue is potential pollen or microspore mother cell. Potential pollen or MNC, microspore mother cell. Okay, so the microsporangium are arranged. Microsporangium are arranged in cluster of 4 cells. Okay, in cluster of 4 cells. In cluster of 4 cells. As the anther matured, when anther mature and dehydrate, the microspore dissociate. Okay, see, as the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grain. When the anther mature and dehydrate, all the microspore is present here. Okay, microspore. Okay, this microspore dissociate from each other and develop into pollen grain. And pollen grain are formed and released, and this pollen grain is released with dehiscence of anther. With dehiscence of anther. Okay, I told you earlier also. See, as the anther develop, the cell of sporogenous tissue undergo meiotic division, and then it forms microspore tetrads. Okay. When anther develop, then cells of sporogenous tissue, okay, sporogenous tissue undergo meiotic division and form microspore tetrad. Microspore tetrad. Okay. The microspore, this microspore tetrad, it contain microsporangium. This is microsporangium. Okay. Microsporangium. Okay. This microsporangium, as when anther mature, ठीक है, okay, when anther mature, this microspore dissociates from each other and develop into pollen grain. And pollen grain are released with the dehiscence of anther. Okay. So with this, the first part of this chapter is over. Okay. We'll continue this chapter in the second video. Thanks for watching. Thank you so much.